A very good morning, dear students. I am Dr. Ravi. So today I'll be discussing about biotechnology and its application. So you would be uh, knowing the definition of biotechnology already. So biotechnology is defined as utilization of living organisms, the products and its processes for the benefit of mankind. So there is also another definition of European Federation of Biotechnology, which we have studied in biotechnology principles and processes. So biotechnology has huge applications in biopharmaceuticals. You know that there are many companies like Biocon in Karnataka and others who are producing the pharmaceutical drugs utilizing the genetic engineering. Then therapeutics. So in India, we nearly have around, we are using around 12 types of therapeutics. Therapeutics means providing remedies. Then there are molecular diagnostic tools, so which are used for diagnosing the diseases. So again, it is utilizing the knowledge of biotechnology. Today, if there is an early detection of diseases, that is because of the progress in the biotechnology. So even HIV is identified very early. So even during the corona period, we also noticed that there was an early detection of corona in patients by using this RT-PCR technique. So if any one of you has undergone that, so the test for corona during those days, you can remember that. So PCR is polymerase chain reaction there. Then genetically modified crops. Some of the genetically modified crops that have had a huge impact on us are the golden yellow rice. Even though it was successful, commercially it has not been successful. People haven't accepted this golden yellow rice. If we were utilizing golden yellow rice, we could have overcome this VAD. VAD is nothing but vitamin A deficiency. So many of the processed foods also utilize the knowledge of biotechnology. Then bioremediation, like if there is an oil spill in oceans from the tankers, ships, or due to war, someone might have let out the crude oil into the oceans. So they can be degraded by using microorganisms. So you might have already known about the organism Pseudomonas butida, which can be utilized for that. So bioremediation, that is providing remedy for such oil spillage and other things, is possible because of progress in biotechnology. So today we are playing with the organisms, manipulating the organisms as per our requirement. The waste treatment. So one of the biggest difficulty that human society and any civilized society has is the treatment of solid waste and other things. So we can utilize microorganisms for biodegradation of these sort of waste, both domestic waste as well as industrial effluents. Then the concepts of biotechnology can also be utilized for energy production. So we can utilize it for the energy production, utilizing the genetically modified organisms. And it can also be utilized for the production of or enhancing the starch fuels like ethanol. Today, there is a talk about ethanol being mixed with fuels to have more efficiency and lesser cost of fuel. So the three critical areas of biotechnology is where research is frequently done is providing the best catalyst and creating optimal conditions for this catalyst to act. And the third important area is the downstream processing. So you can also see the downstream processing where there is an isolation of the recombinant proteins from other impurities and as well as purification of this recombinant protein, or we also call it as desired protein. So the applications in agriculture have been varied. So agrochemical based agriculture, organic agriculture, genetically engineered crop based agriculture. So I've come across many questions where in our books wherein the improvement of food can be by any of this method. But even though predominantly at present 80 to 90% of our entire agriculture is agrochemical based agriculture. So organic agriculture is gaining its ground, but not as effective as agrochemical based agriculture. Genetically engineered crop based agriculture will be the near future solutions for improving the food production. If there is a question regarding this, like improvement of food production, all the three plays a role in improvement of food production. So in our NCERT book, they have not given as to which is going to enhance or which has upper hand in the production of food. So there is no such comparison. But in the real fact is the agrochemical based agriculture is having more popularity at present, and it is nearly 80 to 90% of the entire world is based on food production is based on agrochemical based agriculture. So what are genetically modified organisms or transgenic organisms? So as you already all know that genetic engineering is manipulation of genes to obtain a desired product. So if we are requiring vitamins or antibiotics or enzymes, so we can modify the organisms by recombinant DNA technology and obtain the desired product. So the genetically modified organism can be defined as organisms whose genes are altered by manipulation. So especially during the recombinant DNA technology. So that is how we obtain genetically modified organism or transgenic organism. I was going through a question in our one of the NEET books wherein they had asked, so organisms which are genetically engineered are called as transgenic organisms, or they had even asked it as transformed organisms. Now transgenic organisms would be more appropriate because genetic engineering implies utilizing the recombinant DNA technology, and it is a recombinant DNA. Recombinant DNA is a combination of vector as well as the alien DNA. So whereas if we are talking about transformed cells are of two types, the recombinants and non-recombinants. Transformed cells with recombinants and transformed cells which are non-recombinants. So the answer would be partially right. So the organisms whose genes are altered by manipulation or genetic engineering, so which has a foreign gene, the appropriate answer would be transgenic organism. So transformed organism is partially right because transformed organism can also possess just the non-recombinants without the foreign gene. So that is how the answer is transgenic organism. The advantages of genetic modification in plants. Now if you see this points here, so how could the question be framed in our NEET examination? So one of the favorite examiner questions that I have been noticing from past 3 years is incorrect statements. The major reason that they go for incorrect statement and other things is to see to it that students spend more time in reading the question, and the paper should be of 90 minutes duration because most of the students complete their paper within half an hour. So they make you to read, at least it takes you 30 seconds to read the question. After that, analyzing and identifying the wrong statement, it takes time. So you can expect more number of incorrect statements, then correct statements, assertion and reason type of questions, match the following, and statement-based questions in the upcoming NEET examinations also. So the points that are here, the advantages of genetic modification in plants are, we are going to obtain crops which are tolerant to abiotic stresses. Abiotic stresses implies water scarcity. It can be the nutrient scarcity, unavailability of proper temperatures, or all these comes under abiotic stresses. You might have already studied it in plant growth and growth regulators. One of the stress hormone is abscisic acid. Abscisic acid helps to reduce the abiotic stresses. So you can just recollect that point also here. The pest resistant crops. Now the word pest is a general word. This pest can include bacteria, virus, or it can even include the fungi and nematodes, insects. So whenever we say pest resistant crops, it can be resistant to the insects. So most of the BT plants, they are resistant to insects. Then we have also understood the nematode resistance in plants, especially tobacco plants, using this RNA interference strategy. So all these are the pest-resistant crops can be obtained by genetic modification in plants. Either it can be BT plants, some of the examples like BT cotton, BT corn, or it can also be the nematode resistance in tobacco plants. They are all obtained by genetic modification. Reduce post-harvest losses. What it implies is after post-harvest losses means the shelf period for storage of these fruits or vegetables or crops that we have obtained is for shorter duration. So most of the genetically modified plants, they can have a longer shelf life. That is, you can store them for longer duration. So that is one of the possibility of this genetic modification. Increase efficiency of mineral usage. So this is also one of the things that can be inculcated in a plant if we are going to add the genes so that they have a better efficiency of mineral utilization. It enhances the nutritional value of the crops or food. So that is seen in golden yellow rice. So the other advantage of this biotechnology in plant cultivation is we can obtain tailor-made plants to supply alternative resources for industries like starch, fuel, and pharmaceuticals. So we can obtain alternative resources apart from the regular resources like starch, rich source of starch. You can obtain it from other plants apart from potato is a very rich source of starch. Or fuel plants, biofuel plants, there are a few of them like jatropha or pongamia. So you can also modify other plants to obtain the fuels. Corn is also being used for extraction of ethanol, which is also used as a fuel. So we could utilize other alternatives by the genetic modification and we can obtain fuel from them. So these are the advantages of the genetically modifying the plants. The pest-resistant plants, as I've already told you, pest is a general word. It includes bacteria, fungi, viruses, insects. It even includes the nematodes. Anything which is going to be harming the plants or unwanted organisms which reduces our yield. So we call those as pest. Pest is more like synonymous like nuisance. So these are the pest-resistant plants. We have a few of them. So the pest-resistant plants, it can act as a biopesticide. That is, the plant itself can produce the toxic materials and kill the insects. It reduces the need for insecticides, as in case of BT cotton. So BT cotton, the Bacillus thuringiensis, we have obtained a toxic gene, the Cry1AC and Cry2AB, which can be utilized for controlling bollworms in cotton, and Cry1AB can be utilized for controlling the stem borer. So the BT corn, rice, tomato, potato, soybeans, they are all the pest-resistant plants which prevents the farmer from using the insecticides. Some of the insecticides are non-biodegradable. You already know about the effect of biomagnification of DDT. There is also endosulfan. So many of the insecticides are quite toxic and poisonous. So that reduction of utilizing such insecticides can happen by us obtaining a pest-resistant plants using the biotechnology. So BT stands for Bacillus thuringiensis, the name of the bacteria. It is a rod-shaped bacteria. So Bacillus thuringiensis. So they are going to synthesize proteins that can kill insects like, so the group of them are coleopterans, lepidopterans, and also the dipterans. So here you have three of them. So you can remember them in the same sequence using the mnemonic CLD. So coleopterans, lepidopterans, and you have the dipterans. So they can ask it as match the following. So in column 1, you can have this coleopterans, the group name of the insects. In column 2, they can ask you for the appropriate examples. So you should remember it. When you are reading the paragraph itself, you should understand that they can frame it as the match the following questions. Now Bt toxin, that is the Bacillus thuringiensis toxin, the toxic insecticidal protein, which is in crystal form, that is obtained from Bacillus thuringiensis, we call it as Bt toxin. Toxin means poisonous substance. So Bt toxin does not kill the bacillus. So why does it not kill the Bacillus thuringiensis? Because it exists as inactive protoxins. Just like our enzymes being secreted within our body as inactive enzymes to prevent the cells which secrete them from being digested, even in the Bacillus thuringiensis, the Bt toxin is secreted in as inactive protoxins. So they are activated only in the alkaline pH. So the Bt, this they can frame it as assertion and reason question. So when you are reading the paragraph, you should also be able to analyze what type of question they could ask. So this can be framed as assertion and reason. So Bt toxin does not kill the Bacillus thuringiensis, which is secreting it. That is assertion. Reason, it exists as an inactive protoxin within the bacillus. So both A and R are correct, and R is the correct explanation for A. So this type of questions you can expect from it. So whenever there is a because option of it, that can be framed as an assertion and reason question. So the alkaline pH of the gut of insect converts inactive protoxin to active toxin. So this is happening within the insect. So in the insect, we are not using the term the stomach and other things. Even though I was reading in one of the meat books where they have used the word stomach, it is not appropriate to use it. So more of we should be using the terms like foregut, midgut, hindgut. If you have studied the digestive system of cockroach, you can remember that we would be using the terms like crop, gizzard. So these are the terms that we use. So we don't use the term stomach in case of insects. So anyway, the right word would be alkaline pH of the gut of insect converts inactive protoxin into active toxin. So the alkaline pH is required. The pH which is above 7. So Bt toxin is coded by a gene and we call that as CRY. See, the Cry represents the crystalline nature of the Bt proteins. They are crystalline in nature. So Bt toxin is coded by a gene which is named as Cry. So Bt toxins are insect specific. So if you are looking into these Bt toxins, they are insect specific or insect group specific also. I have already told you there are 3 groups of them. So the coleopterans, lepidopterans, dipterans. So now the cotton bollworm is controlled by 2 Bt toxins. One is Cry1AC, Cry2AB. So in order to remember in this systematic way, I always give to my students the words like if you cry for the first time, you get AC. If you cry for the second time, you have to develop apps. So that is how I ask them to remember this 2 of them. So Cry1AC, Cry2AB. So these are the 2 genes which controls the cotton bollworms because they are going to produce the toxins, Bt toxins, which acts at the pH part of their gut. So, and they are going to make a hole into the insect's gut, and it's a very painful death for the cotton bollworms. So in corn borer, the Bt toxin is Cry1AB. So we should be very much thorough with these points, which can be asked either as match the following or as memory-based question in the NEET examination. So the next one is the nematode resistance in tobacco plant. So nematode belongs to the Ascalmenthes, that is the roundworms, and it infects the roots of tobacco plants. The host plant is tobacco plant. Scientific name is Nicotiana tabacum. This they have given in the Solanaceae family as fumigatory plant. So they are also they produce this carcinogenic substance nicotine, which you are aware about. So they belong to Solanaceae. So you just know some information apart from what has been given in the NCERT book. The host plant is the roots of tobacco plant, Nicotiana tabacum solanaceae. So the pathogen is Melidogyne incognitia. This is a nematode. So Melidogyne incognitia is a nematode which belongs to the phylum Ascalmenthes. So what is the strategy that we are having to control this Melidogyne incognitia and prevent it from affecting the tobacco plants? The strategy that we are adapting is RNA interference, or we also call it as RNAi control, where RNAi means ribose nucleic acid interference. So RNAi control or RNA interference strategy is naturally occurring in many of the eukaryotes as a cellular defense. We have just copied it from that and we are utilizing it in our control of this nematode Melidogyne incognitia. So it's a cellular defense mechanism in most of the eukaryotes. So this RNA interference, so what it is going to do is they prevent the translation of a specific mRNA due to a complementary double-stranded RNA. So the double-stranded RNA breaks this specific mRNA into fragments. And once they are broken down, so these points are not given in the book, but if you refer the reference books, you will be obtaining those information. So they are going to break the mRNAs and thus prevent them from undergoing the translation process. And you know that if there is no translation, so there is no protein. And some of them are very crucial proteins which are required for the survival of nematodes. So when these crucial mRNAs are destroyed due to the presence of this double-stranded RNA, it is going to be lethal for the nematode and the nematodes are killed. This is what we are following it as the RNA interference strategy. See the DNA for complementary double-stranded RNA. So that nematode-specific gene, what we call it as, it is obtained from RNA viruses or it is also obtained from mobile genetic elements. So we call it as nematode-specific genes. So this topic, if you try to remember through this chart like with keywords, it would be easier for you. So it's better you write the keywords and try to recollect them. So nematode-specific genes or DNA which can produce double-stranded RNA, it is obtained from RNA viruses or from mobile genetic elements, which we call it as transposons. Don't go into too much of depth of this mobile genetic elements or transposons. They are also called as jumping genes. For the first time, this was work by Barbara McClintock in maize plant. And she got Nobel Prize for this jumping gene or the effect of understanding the mobile genetic elements or transposons in maize. So you can just remember this additional point. So the source of this nematode-specific gene is RNA virus or the mobile genetic element transposon. Now we have to make a recombinant DNA. The recombinant DNA includes Ti plasmid plus nematode specific gene. So in our book, NCERT book, the Ti plasmid is instead given it as Agrobacterium vector, and so we cannot use PBR 322 or PUC 18 for plant experiments. So we are using the Agrobacterium vector Ti plasmid plus nematode specific gene. So that is going to form the recombinant DNA. Now this formed recombinant DNA with nematode specific gene, we have to introduce into the host cells, that is the tobacco plants. And we can follow this method of biolistics or gene gun. So you know that the introduction of, there are three methods of it, like microinjection, biolistics or gene gun, then disarmed pathogen vectors we can utilize. So here biolistics or gene gun is frequently utilized for the plant cells, introduction of recombinant DNA into the plant cells. So they are introduced into the host plant, this recombinant DNA with foreign gene, that is nothing but nematode specific gene. So this nematode specific DNA, they can produce double-stranded RNA. So that we should be aware about. So why they are able to produce double-stranded RNA is both the template strand as well as the coding strand produce the RNA. So they produce sense RNA and antisense RNA. Since sense RNA and antisense RNA are complementary, they become double-stranded RNA. So this is introduced into host plant, this recombinant DNA by biolistics or gene gun, and it becomes the transgenic tobacco plant. So they produce this double-stranded RNA. The reason is by they produce the sense and antisense RNA in the host cell. So as a result of it, since they are complementary, they form the double-stranded RNA. So this initiates RNA interference. So the specific mRNAs are broken down by the double-stranded RNA, and they are silencing the specific mRNA, which is crucial for the survival of the nematode. So once this specific mRNA of nematode is silenced, so the nematode is killed, and that is how the transgenic tobacco plants avoids the nematode. So these are the key points that you should be writing down in a chart, like so to remember the points better. So, and many questions can be framed from them during NEET. So the applications in medicines. So that is the next concept that we have in biotechnology and its application. So recombinant DNA technology, it is a safe and effective mass production of therapeutic drugs. So the therapeutic drugs, they are utilized to provide remedy. So this drugs that are obtained from this recombinant technology, they are mass produced. Like if you see in Biocon, which is very near to us in Bangalore, so they are producing en masse large amount of insulin. So using this recombinant DNA technology as well as a modified the bacteria host cell, that is a transformed cell. So the effective mass production of therapeutic drugs. So it does not, what is the advantage of having a therapeutic drug is, it does not induce unwanted immunological responses, no rejection of the medicines. So 30 recombinant therapeutics have been done throughout the world, and this information, most of them are updated during 2013 or 14 when they had the syllabus. The number of recombinant therapeutics might be more or lesser. So, but you should be just aware about that. So it is data that they have given long back. So only 12 recombinant therapeutics have been approved in India and being marketed in India. So this can be asked as fact-based question. So the next part is after recombinant therapeutics, genetically engineered insulin can be obtained using this biotechnology knowledge. So the earlier days, they were using the pancreas of animals to obtain insulin, and it caused allergy. But nowadays, we are using the genes to synthesize chain A and chain B separately, as it was done by this American pharmaceutical company, The name of that American pharmaceutical company is Eli Lilly. So they produce chain A and chain B, which are linked by disulfide bonds later. So they produced it separately and they linked it later to form the disulfide bonds. In mammals, you find that the insulin will be secreted as prohormone. That is, it will be secreted as proinsulin. If you look into the structure of proinsulin, so they'll be having C chain which connects the A and B chain. So this is seen in case of mammals. Proinsulin is synthesized, and when it is undergoing maturity, the C chain is removed from A and B chain. So A and B chain are held together by two disulfide linkages. One of the disulfide linkages is in the A chain. So if you study the structure of insulin as given in your textbook, you can notice all those features. In mammals, proinsulin is synthesized, which becomes mature only after removal of the C-peptide chain. So this is what is the process that is naturally occurring in our body. But in the American pharmaceutical company Eli Lilly, what they did was they synthesized chain A and chain B separately using this recombinant DNA technology. So two cultures were done separately, and later they were linked by this disulfide bonds. The next aspect that we go into is gene therapy. So gene therapy, the definition is it's a method of correcting gene defect diagnosed in a child or embryo. This is what is the definition that is given in your NCERT. A method of correcting gene defect diagnosed in a child or embryo, we call it as gene therapy. Still now they have done gene therapy for two of them. One is cystic fibrosis and another is we have the SCID. This name is not given. Instead, in our books, they give it as ADA deficiency, or it is also called as bubble baby syndrome. So this is what you should know about the ADA deficiency that we'll be studying. ADA means adenosine deaminase deficiency. And cystic fibrosis and SCID are one in which gene therapy has already been worked. See, the present medication system that we have is where it's a chemotherapy, where the chemical substances are prescribed either in the form of tablets or capsules or as syrups or as injections. But in the near future, gene therapy might be a method of the providing solutions for at the root level and rectifying the root cause of any of the disorders. So as of now, you should remember these two examples in your textbook. So ADA deficiency has been covered, adenosine deaminase deficiency. So what is SCID? Severe combined immunodeficiency disorder. So it is similar to AIDS, except differences in disease are different, but what is AIDS? Acquired immunodeficiency syndrome. So here, severe combined immunodeficiency. So in both of them, there is immunodeficiency. So the patients who are affected by these disorders, they do not have immunity. And they are like a walking culture media for all the pathogens. So that is why they do not have resistance against any pathogen and they are affected by pathogens. So that is because of the deficiency in immunity. So most of these patients who are affected, especially the young infants, they are protected within a bubble. That is why we call it as bubble baby syndrome. So the first girl who got the gene therapy for ADA deficiency is Ashanti D. Silva, who is 4 years old girl. She was the first patient to be treated for this gene therapy for ADA deficiency, or you can also call it as SCID or bubble baby syndrome. So SCID is severe combined immunodeficiency adenosine deaminase. That is what ADA is. It's a enzyme. Whenever this enzyme is absent, it is crucial for the functioning of immune system. So if it is absent, the immune system doesn't function. That is why the patients suffer from severe combined immunodeficiency. The cure for this is by bone marrow transplantation or enzyme replacement therapy. So bone marrow transplantation, especially we are talking about red bone marrow. So bone marrows produce the RBC and WBC. See, the major ADA gene is seen in lymphocytes, particular cell lymphocytes. So white blood cells, we call it as the leukocytes. White blood cells, you know that they are of two types, granulocytes, agranulocytes. So you have under agranulocytes, monocytes and lymphocytes. Monocytes are the largest cell. Then you have the granulocytes like acidophils or eosinophils, basophils, neutrophils. So based on their staining ability. So based on the percentage of distribution, the total number of WBC percentage of distribution, neutrophil is found in large amount. These are all facts that you already know about. So bone marrow transplantation, so where they are going to, if bone marrow is transplanted, so they might be producing the normal WBC, especially the lymphocytes with normal ADA gene. That is why they go for this bone marrow transplantation. Or it can be enzyme replacement therapy. That is the injection of functional ADA into the patient. So bone marrow transplantation, enzyme replacement therapy, they are all temporary and they are not permanent. So, but if you want to have a permanent cure in the patient, so the ADA gene introduce that early embryonic stage in cells, it could be a permanent cure. Whereas frequent treatment has to be done whether it is bone marrow transplantation or enzyme replacement therapy. So this is possible because of the technique of gene therapy. So let us understand what are the process involved in this introduction of it, this ADA gene. So lymphocytes from patient's blood are separated and they are in vitro. In vitro means in the lab conditions. The functional ADA cDNA. Whenever we are using the word cDNA, you should understand that it's a product of reverse transcription. That is, we have obtained it from mRNA. We have subjected them to reverse transcription and obtained them. cDNA is nothing but complementary DNA. So the functional ADA cDNA is introduced through retroviral vector. So we are using the disarmed retroviruses for introducing this functional ADA cDNA into the separated lymphocytes. So now the lymphocytes are modified and they are having this ADA cDNA. They are returned back to the patient's body. This process should be frequently repeated as the lymphocytes are not immortal cells and they have the expiry period is there, or their lifespan is not for long duration of time. As a result of that, they are frequently, this process is repeated to keep the patient in normal conditions. So the lymphocytes are taken out from the body, and within the lab, we are going to introduce this ADA cDNA into the lymphocytes, and this modified lymphocytes are returned back to the patient. So the process should be repeated frequently as they are not immortal cells. Then we go for the next part of it, that is the molecular diagnosis. The early diagnosis of diseases can be done by this genetic engineering or by our knowledge of biotechnology. So one of them is recombinant DNA technology. This technique can be used for early detection of diseases. PCR, polymerase chain reaction, we have read more in detail during the biotechnology chapter. Then ELISA, enzyme-linked immunosorbent assay. So these are all the methods of early diagnosis of the diseases. So if we diagnose early, the treatment could be started at a very early stage, and we can increase the longevity of the patient and sometimes the cure can also be given. So very low concentration of a bacteria or virus can be detected by amplification of the nucleic acid by PCR technique. So you know the three steps involved there, which I call it as DAY. So that is denaturation, annealing, and extension. The PCR used for early detection of HIV in the AIDS patient. So this is preferred because they can detect the nucleic acid and amplify this pathogen's nucleic acid. So we can detect it quite early at early stages of infection. So especially this played a very crucial role in stopping the spread of coronavirus during the COVID times. So to detect mutations in suspected patients of cancer, so these PCR are used. The PCR is also used to detect many genetic disorders. So mutations. So when we are talking about mutations, how do we identify them? See, usually the single-stranded DNA or RNA, so they are tagged with radioactive probe. It's a known sequence. So this hybridizes, the DNA hybridization happens, and the cDNA, they are subjected to the auto-radiation. Okay, students, so there was this, the clone having mutated gene, they are not seen on plate. Why they are not seen on plate is they do not have, the probe does not have complementary sequence to the mutated DNA. As a result, they won't tag with the DNA having mutated gene, because of which they are not seen on the photographic film. Whereas the clone without mutated gene, they are seen on photographic film. The reason is they don't have any mutations, so the DNA has a complementary base pairing with the tag. So that is how they are seen on photographic film. So that is how you can differentiate. So if the mutated genes, they are not seen on the photographic plate, whereas the normal genes are seen on photographic film. So probe has no complementarity with the mutated gene. So they can ask it for assertion and reason. So they can ask assertion as the clone having mutated gene is not seen on the photographic film. So reason for that is probe has no complementarity with the mutated gene. So which implies both A and R are correct, and R is the correct explanation for A. So I'm just framing right answer questions for assertion and reason, but you should be prepared for this. The options can be anything. So the next diagnostic method is ELISA method. ELISA is based on antigen antibody principle. You already know that ELISA is enzyme linked immunosorbent assay. The infection by pathogens can be recognized either by studying the presence of antigens. You can identify the antigens and you can say that the disease has happened. Or another, the indirect method is you identify the antibodies synthesized within the body and you say that these antibodies which are specific to a disease, so as a result of that, the person is suffering from the particular pathogen. So this is how you can use this antigen antibodies to identify the disease. Directly you can identify the disease by the presence of antigens, which will be having proteins or glycoproteins, and indirectly you can identify the pathogens by detecting the antibodies synthesized in the patient's body against the pathogen. So the next topic that we have is transgenic animals. So the alteration of animal genome by introduction of foreign gene by manipulation, we call it as transgenic animals. So transgenic animals have a foreign gene and they have altered their phenotype. So the animal genome is altered by the introduction of a foreign gene by manipulation, in the sense recombinant DNA technology. So 95% of transgenic animals are transgenic mice. So that we have to remember about. The benefits of transgenic animals are, so to study normal physiology and development, like insulin like growth factor. So we can study the normal physiology and development using these transgenic animals. So insulin like growth factor is the example that they have given. So to study the contribution of genes in the development of a disease. So here you can understand the progress of a disease and its nature of a disease. By using the transgenic animals, we can study the contribution of genes in the development of a disease. The biological products can be obtained using transgenic animals. So transgenic animals obtaining products like antibiotics or antibodies, they are cheaper than through the microorganisms, because producing culture media, maintaining a sterile atmosphere is going to be very expensive. So as a result of that, the biological products, especially we would be discussing about the alpha-1 antitrypsin. This antitrypsin is used for treatment of smoker's lung or emphysema. Then we are also using this, the rosy cow, the transgenic animal from which alpha-lactalbumin, human lactalbumin is obtained from this rosy cow. The chemical safety, the vaccine safety testing also, we can use transgenic animals and avoid the monkeys. So that is one way we can have much more effective study by using transgenic animals to test the efficiency and effectiveness of vaccine rather than using the monkeys. So the chemical safety testing can also be done using the transgenic animals. So we can identify even at very low dosages the toxicity by using transgenic animals, whereas normal animals, they wouldn't be showing that sensitivity to chemical toxicity, but transgenic animals would be showing a better result than the normal animals, especially for chemical safety testing or the toxicity testing. We can utilize the transgenic animals. So these are the places where the transgenic animals are utilized. Ethical issues, the problems of unpredictable results, genetic engineering approval committee. So if you have seen most of the superheroes, they're all mutants, whether it is Ninja Turtle or Spider-Man who was bitten by a mutant spider. So they're all examples of the genetic engineering having gone wrong. Even though it is fiction, so in reality they are all possible. So the problems of unpredictable results are there. So and also we cannot use human beings as lab rats and conduct experiments. So for this, the government of India has the Genetic Engineering and Approval Committee who regulates the experiments being conducted on. Also an approval from them is required for conducting the biotechnological experiments. So and also there are the problems of patent. So basmati rice, 27 varieties of basmati rice we already have traditionally being grown in India. But in 1997, one of the American company got patent rights through US Patent and Trademark Office. Sometimes these years are asked. Last year they had asked about the year in which one of the treaty was signed, probably I think Montreal Protocol. So remember the years also. So even though it is not a standard questions to be asked, which is more of history type of question, but last year they had asked a question based on year. So try to remember the year also. So the 1997, an American company got patent rights through US Patent and Trademark Office, wherein they had crossed Indian basmati variety with semi-dwarf varieties, and they got a variety which was producing basmati rice, and they applied for the patent. So this is, if they get it patented, then we would not be able to grow those. So there is biopiracy also, the use of bioresources by multinational companies without proper permission or authorization from the countries and people concerned. So there is a big difference between developed countries. Developed countries have a very good, they have a very good amount of industries and money, whereas the developing and underdeveloped countries, including India, we have a rich biodiversity and traditional knowledge, but we do not have that economic strength. So this is what we have to remember regarding the reason for the exploitation of many of the MNC companies, especially the developing and underdeveloped countries, which are rich in biodiversity and traditional knowledge. So the Indian government came up with an amendment for Indian Patents Bill. So, and they regulate the patent terms, the emergency provisions, and research and defense initiatives. So these are being done. So this is a brief synopsis of the biotechnology and its application chapter. So I'll be discussing in the next video part of it 25 questions, which would be interesting questions concerned with biotechnology and its applications. Thank you. Join me in for the second video.